At this point, you already know that DNA replication starts in the S phase of the cell cycle. And in mammals, the S phase can take around 8 to 12 hours to complete. In textbooks, typically the chromosome is represented as this bar-shaped thing. But this is really a condensed chromosome, which is seen in mitosis. For interphase, or S phase, the picture is not so pretty, and the DNA, or chromosome, is a loose hairball with no special structure. This is called the chromatin, and it is a non-condensed version of the mitotic chromosome. If we stretch out this hairball, the chromatin may look like a string, and this is our reference point for this discussion. If you remember from the initiation video, there are these helicase double hexamers which license many parts of the DNA for replication initiation. This happens in the late G1 phase, or very early parts of the S phase too. A subset of the chromatin of about 3 to 4 potential replicons, which are these licensed double hexamers, form a domain of about 400 to 800 kilobases. This domain is called a replication domain. Just for clarity, this double hexamer is one potential replicon. This way you can partition the entire chromatin, or chromosome, into multiple replication domains. When it comes to the timing of replication, let's say RD1 starts at time 0, then RD2 starts at time t1. Now, t0 and t1 are almost similar in time, but they're never exactly the same. Then RD3 can have a start time of t2, and it can be t0 as well. T1 and T2 are similar but not identical. Then RD4 can start at time 0, T1, or time 3. The pattern here to observe is that the adjacent domains are never starting the replication initiation at the same time. If that was the case, then you cannot partition, and it would all be one single domain. Another crucial layer of division is if you combine like 2 to 3 replication domains. This forms a CTR, which stands for Constant Timing Region. This way you can have many CTRs along the chromatin composed of multiple replication domains. Just like the replication domains, the adjacent CTRs are never starting replication at the same time. Since there are multiple replication domains in one CTR, they are multi-megabases in length. Typically, we tend to think of CTRs organized into patterns of heterochromatin and euchromatin. And the boundary of each CTR is called a timing transition region. In a moment, I will add details to all this jargon. But the main point to realize now is that the entire chromatin or chromosome is split into short regions of domains that start DNA replication at different times. Let's quickly add more details to all of this. The replication domains contain 3 to 4, sometimes 1 to 4, origin licensed replicons. And adjacent domains start nearly at the same time, but not at the identical times. CTRs are the main players, which contain multiple smaller replication domains, which means that the CTR can be multi-megabases in length. This leads to the point that all replication domains in a CTR are starting replication at nearly the same time. Hence the name Constant Timing Region. Let me expand on this. Say RD6 and 7 are in the same CTR. Then RD4 and RD5 are in the same CTR. In this example, the replication domains within a CTR are similar to each other, but RD5 and RD6 are very far apart in their timing. This leads to the point that CTRs perhaps represent a single unit of replication starting time. At some level, they correspond to domains of gene activity, which are heterochromatin and euchromatin. If you are new to these things, this may be a lot of information to take in. So let's take a step back and try to see why it may be useful to partition DNA replication timing. Since this is an active area of research, the answer is still unclear. It is, however, thought that CTRs prevent or limit the licensed replicons to fire. That is a way to control resources involved in DNA replication, like proteins and factors involved in this process. The other answer could be that the timing is actually a feature of the space in which the DNA is physically organized in the nucleus. Again, these are plausible answers to a process which is still unclear. Let me explain this space and time of DNA replication through a simple sketch. If you look at a nucleus, you have two main components to consider: the nucleolus and the nuclear membrane, which contains lamina and the cytoskeleton of the membrane. Since S-phase DNA is a loose hairball, each chromosome in the nucleus is present in a constrained space of its own. There is little mixing of chromosomes, and this we call the chromosome territory. Now you see that there are nucleolar and lamina-associated parts of any particular chromosome. We call them to form B compartment, which is another name for heterochromatin, which is relatively inactive at the transcriptional level. This would be the late CTR portion of the chromosome. The middle portion here is the active region, or A compartment, also called the euchromatin, and this would be the early CTR. The area at the edge of late and early CTR is what we call the timing transition region, which defines the transition between early and late CTRs. This is a simplified view of how space impacts the timing of DNA replication initiation. There's a list of potential range of factors that regulate DNA replication timing. We will discuss 7 of them. First, RNA. Specifically, link RNAs, also called long non-coding RNAs. Sometimes even RNA from transposons can regulate DNA replication timing. The clear example of link RNA is the ZIST or ASAR link RNAs. The ZIST RNA codes one of the X chromosomes completely, which leads to X chromosome inactivation in females. This also makes the inactive chromosome a late replicating chromosome. This is a special case where the entire chromosome is one giant late CTR because of ZIST. The ASARs are slightly different. They code specific parts of the chromatin, and those parts where ASARs localize become late CTRs. This is a correlation, but the paradox is that if you delete these link RNAs, the late CTRs actually become extra late CTRs. So it is unclear how the link RNA actually affects replication timing. Second, DNA. A special case of this is the locus control region, present in the beta globin locus. This control region makes the locus an early CTR. This special DNA region is conserved in its CTR function in both humans and mouse. And you can convert a late CTR to any early CTR simply by inserting this locus control region into a late CTR. Third, which is also DNA, but a more general case called early replicating control element, or ERCEs. These look like enhancers in transcriptional regulation because they bind a bunch of master transcription factors. And you could say that locus control is also an ERCE, but not all ERCEs can convert a late CTR to an early CTR. They're also different from the special case of beta globin locus in that you need at least 2 ERCEs in a CTR to make it an early CTR. Just a side note, these CTR boundaries called TTRs overlap with another feature of the genome organization called TAD boundaries. I will cover this when we get to the genome structure and organization. Moving on, the fourth one is the physical proximity to lamina or nucleolus, or any heterochromatic feature in the nucleus. We already went over this, that proximity to lamina makes chromatin a late replicating region. However, this is not a rule, and I have point 5 to explain why. The timing decision point, which is a mystery feature that defines where a piece of DNA should be located in the nucleus when starting the replication in the S phase. TDP typically works in the G1 phase, so it defines the early CTR. And now to the exception to point 4. Say TDP defines that this portion here starts replicating early, but over time this segment of DNA diffuses into the lamina. Since it already started replicating, this early CTR will not become a late CTR. Likewise, a late CTR moving out of lamina will not become an early CTR. Sixth factor is epigenetics. Classic example is DNA methylation, which is seen on ribosomal DNA locus. There are multiple copies of our DNA in mammals spread across many chromosomes. Some of them are early and some of them are late CTRs. All the late ones have a higher DNA methylation on them. This is because the methylation is a target of nucleolar remodeling complex, which has a silencing effect on the locus, which converts some copies into heterochromatin, which is also what we call the B compartments. As a result, they become late CTRs. The early ones have low or no DNA methylation, so NORC cannot bind them, no silencing happens, and they're active and remain as early CTRs. The ZIST lincRNA we discussed earlier, which converts the X to an inactive X, also works through epigenetics, where it recruits polycomb repressive complexes to do the inactivation. We talked about PRCs in the epigenetic replication video, so check that out for more details. In addition, we also have some correlations that euchromatin has features like H3K4 methylation, H3K27 acetylation, and many more that correlate with early CTRs. In contrast, the heterochromatin has repressive marks like H4K20 methylation, which correlates with those regions being in the late CTR. Example is this NORC silencing effect, which is mediated through this H4K20 methylation. Alright, the last point is about controlling the origin firing directly. We saw in the initiation of replication videos that licensed origins contain a double hexamer of helicase in the late G1 phase. And then firing starts when a DDK complex comes in and phosphorylates the double hexamer. And then the licensed origin assembles into a CMG helicase. Simple way to stop this is to have a blocker of DDK activity, and TAS1 proteins do that, which delays the firing of the licensed origin, and you get a late CTR. Alternatively, you can start removing the phosphates from the double hexamers. Proteins like RIF1 and PP1 do exactly that, and delay the overall timing of the origin firing. Conversely, you can also reduce the DDK recruitment time. SWI6, FKH1, and CTF19 can exactly help in that. This converts such regions into an early CTR. The area of space and timing of DNA replication is still relatively very new, and many things remain to be discovered. But hopefully this video and discussion gives you some idea about it.